Hello, everyone, and welcome to the Human Cell Atlas Biological Network Seminar featuring the Adipose and lung networks. So, the seminar series goals are to increase the biological network visibility and to spark new opportunities for collaboration and engagement, to promote coordination with other networks such as the CZI, seed networks, to lay the groundwork for drafting Atlas roadmaps in each biological area in preparation for work sessions at the virtual 2021 June HCA General Meeting. And this seminar series takes place the second Thursday of each month, 10:30 to 12 p.m. Eastern Time. And we are very grateful to the Chan Zuckerberg Initiative for their generous support to make this seminar series possible. So we ask folks to please submit their questions using Slido. So if you go to slido.com/HCA Bio Network, you can submit and vote on questions. You can also submit and vote on questions by scanning this QR code at the top right corner using your phone. So you can submit a question at any time, and if you just go to the Q&A at the top panel, that's how you will submit the questions. And just to let everyone know, we do encourage you to vote on questions. Questions with the most votes will be prioritized. So we would like to hear from you. So we're going to open up a very quick poll here. So please go ahead and go to Slido by scanning that QR code or typing in slido.com/HCA Bio Network. And the first question in our poll is, what would you like to get out of this seminar? So I'll give everyone a couple of seconds to let us know what's on their mind. Okay, so I'm seeing knowledge, I'm seeing learning about the biological networks, new collaboration, new information, updates in the field. These are all great. Okay, keep those questions coming in. So the next question that we have here, and this is really important because this is how we can reach out to you and how we can best connect with our collaborators. So how did you hear about this seminar? It looks like direct mail and Facebook are neck and neck. Great. So I'm going to give everyone another 10 seconds or so to complete this. Looks like direct mail is still the best way to reach people. Perfect. And then our last question in the poll is, are you part of any of the HCA biological networks? And it looks like 64% of you are so far. And to let you know, for those of you who are not part of any of the biological networks, in our closing survey for the seminar, we will have a way for you to let us know if you would like to be part of any of the biological networks, and then we'll get in touch with you after the seminar so you can join up with any of our biological networks. Great. So switching back. So the seminar format for today is we're going to open up with a talk on the Adipose network from Christian Wolfram, and then that will be followed by 5 minutes Q&A, and then we'll go to the lung network, and Nick Banovich will give that talk. We'll follow up with a 5-minute Q&A with him, and then we will go to our breakout sessions. And now I am going to turn it over to my colleague, Ellen Todris, who will introduce our first speaker, Christian Wolfram.
Hi, everyone. We're happy to introduce Christian Wilkin. He's the head of the Laboratory of Translational Nutrition Research at the Department of Health Science and Technology at ETH Zurich. After obtaining his PhD from the University of Münster in 2001, he moved to the Rockefeller University in New York for his postdoctoral training. In 2007, he relocated to Switzerland to establish an independent junior group at the Competence Center for Systems Biology at ETH Zurich, where he was promoted to assistant professor in 2008 and to the associate professor in 2010. In 2014, he became full professor at the ETH Zurich, and since 2012, he's serving as the study director for health science and technology and the Graduate School for Molecular and Translational Biomedicine, and since 2016 as the study director of medicine at ETH Zurich. Christian Wolfram is also the president of the International Conference on the Biochemistry of Lipids, ICBL, and the vice president of the German Society for Lipid Research, DGF. The Wolfram Lab has been focused on understanding adipose tissue heterogeneity and plasticity and its involvement in metabolic control, both at the level of adipose precursor cells as well on the adipocyte precursor cells, as well as on the level of mature adipocytes. Welcome. Christian, please go ahead.
Thank you very much. Thanks to the organizers for the invitation. Can everybody hear me? Yeah, very good. So I'm very glad to present today. I'll talk today about some work we've been doing on especially the mature adipocyte and its plasticity and its importance for regulating, in this case, tissue function. So maybe for those people in the adipocyte field, they're well aware of this, but for the lung people that are maybe half of the audience, adipose tissue is normally today classified into two types of adipocytes. You have the rather white adipocytes, which store lipids. They are highly important to maintain systemic metabolism by keeping the lipids out of the other tissues. And you have, on the other hand, the thermogenic brown adipocytes, which burn energy through the presence of uncoupling protein 1, which basically short circuits the mitochondrial membrane. And this heat production is believed to be utilized, or that it might be utilizable to burn energy and thereby lose weight in obesity, although this is something that we still have to understand. Now, this is a quite simplistic view, both on the level of the precursors, right? White and brown precursors, brown adipocytes and white adipocytes. But actually, since many years, there has been a discussion about a rather more plastic or heterogeneous composition of the adipose tissue, and I'd like to show you some data on that today. So what you see here, this is actually quite a nice example. This is brown adipose tissue at different housing temperatures in rodents. Normally, brown adipose tissue looks a little bit like this here. These are adipocytes with many lipid droplets, which is probably necessary to efficiently liberate the lipids to utilize them as fuels. And you see here in brown the staining for the uncoupling protein 1, which is actually the thermogenic protein that allows the combustion of these lipids. And you can see already that even though some cells have smaller lipid droplets, they have less UCP1 expression, and it's highly variable across the depot. If you now expose an animal to cold, you see the lipid droplets shrink because the lipids are utilized as energy, and UCP1 expression is much more prominent. But even here you can see cells that look very much like brown adipocytes, many small lipid droplets, but some have UCP1, some do not have. So nobody really understands what the reason for this is. And if you put an animal to thermoneutrality, when you don't need thermogenesis, then these lipid droplets increase, and basically these look like white adipocytes, but you still find UCP1 expressing cells even under these conditions, even though these are very few. So we were quite interested in this heterogeneity, and since we are talking about a cell atlas network, we obviously talking about single cell analysis. And I will focus mostly on the latter part here, but a quick explanation. Brown or adipose tissue in general is only to a certain percentage composed of adipocytes. A common mistake, people think adipocytes make up most of the adipose tissue. It's actually between 10 and 30% of an adipose tissue are these lipid-filled cells, and the other 70 to 80% is something else. Fibroblasts, immune cells, endothelial cells, and so on and so forth. So if you want to study the heterogeneity of the adipocytes, you have to somehow mark them. And we did this by using a model that marks the adipocytes with a Cree and induces the expression of a nuclear dye, in this case a red nuclear dye. And then you can sort these red nuclei and use them to perform single nuclear sequencing, either using 10x genomics or SmartSeq2. So in the first approach, this was still with the Fluidigm platform quite a few years ago. You see, we could isolate nuclei. We could actually also generate clusters of nuclei, and it was a bit disappointing, one has to say. So this is a brown adipose tissue of a mouse at room temperature. You have two clusters that are not really segregating very well. This one cluster here was quite interesting, as it is definitely an adipocyte. And based on this expression, it's actually even a brown adipocyte, although it's a bit lower in thermogenic genes. So to overcome this problem with the low number of nuclei, we actually went and employed the more advanced 10x genomics platform. And here you can see a much larger number of nuclei that were analyzed, sorted and analyzed in mice. So these are again nuclei from the mature adipocytes exclusively. This is important. I'll come back later to this when I talk about the human system. You can see here that this now looks quite different. There seem to be multiple populations, even at room temperature, actually 3 of them. A fourth one is a lot smaller. This fourth one was actually this population we also saw segregating a little bit in the C1 experiment. And it's especially marked by this one protein CYP2E1, but also ALDH1A1. The other ones are quite uniform. If you look at the standard adipocyte markers, they express adiponectin, which is one of the most prominent adipocyte-specific genes, but also leptin, and certain thermogenic markers normally associated with brown adipocytes. So to get a bit more insight into these whole populations and to understand whether we are really talking different populations or maybe just different cell states that you pick up in room temperature due to this heterogeneous nature, we did a bit more comprehensive analysis. And what we did is we took brown adipocytes, again, brown adipocyte nuclei, either at mice kept at cold exposure. This was the picture on the very right with the very small lipid droplets, room temperature, or at thermoneutrality. That was actually, it's flipped. This was the picture on the very left with these very large adipocytes that you could see. You can see here that depending on the external stimulus, you get differences in populations if you do a combined analysis. In thermoneutrality, most cells are made up by this, what we call P3. At room temperature, we have a mixture of population 5, 1, and 2. And at thermoneutrality, at cold exposure, this is mostly P1. What's quite interesting is this P4 population. This is present in every single condition, and it seems to increase as the animals are kept at a rather thermoneutral condition. So the lowest amount is here, about 2 to 3%, up to 8% under thermoneutral conditions. So we were a bit more interested to understand about this population because this other population seemed to probably rather be cell states depending on extracellular signaling. As I said, CYP2E1 is a very specific marker, so we moved from the single cell analysis to validate our findings, and it actually turned out to be quite interesting because you see here, this in green is a brown adipose tissue depot. The green marks the UCP1 expression, so it's a transgenic mouse expressing a membrane green dye under the UCP1 promoter. You see these CYP2E1 cells, they appear in the middle of the brown depot, at some of the edges, and they are either multilocular, you can see this quite well here, this is the enlargement from this, or they seem to appear unilocular. You remember in the beginning I said white adipocytes are rather unilocular, while brown adipocytes are multilocular. So we now have a cell type which seems to can acquire both morphologies, and this kind of localization is very difficult to assign to any neuronal, endothelial innervation or whatever status. So quite puzzling, but we were obviously a bit interested what this actually means. So what about other tissues? So when you look a bit more closely at other tissues, for example, the inguinal adipose tissue, you can see that there's quite a few CYP2E1 positive cells to be found. Actually, the number of this type of cell increases with the whiteness of the adipose tissue depot. So in brown, you find about between 2 and 8%, right? In the inguinal, it's about 15, and in the visceral, in the omental adipose tissue depot, which is a really adipose tissue depot that contains mostly white adipocytes, there's about 30 or 40%. What is quite interesting is when you do electron microscopy, and this was done by Saverio Cinti, and you stain CYP2E1 negative or CYP2E1 positive cells, you notice immediately that the mitochondria look very different. So this is a CYP2E1 negative cell, classical brown adipocytes, these beautifully organized cristae, suggesting high mitochondrial activity. And this is how the CYP2E1 positive cells look, right? These highly disorganized cristae, which doesn't really look like these cells are very thermogenic or have a high mitochondrial activity. So this is already gives us a hint that these cells, even though they might appear in the brown adipose tissue, might be thermogenically challenged to a certain extent. As I mentioned, there's other markers you can use. So you see here CYP2E1, ALDH1A1, OUT-S2, and ATP2B4. These are all markers of the of this population 4. And you can actually confirm this. So we used ALDH1A1. This is quite a well-known gene and well-studied. There's excellent antibodies available for this. And if you do a co-staining of CYP2E1 and ALDH1A1, you can see you find these clusters, and the color is highly overlapping. So it's really suggests that we have stained this with 2 markers that from the single cell data seem to be specific for these populations, and you can confirm this quite nicely in these co-stainings. So this is really a population, and these are just 2 of the most prominent marker genes that you can use.
Christian, 2 minutes.
Yeah, so I will quickly talk about the human situation. The humans have this is from brown adipose tissue in humans. Here you cannot sort the adipocytes, and you see here that you get a lot of other cells in the single nuclear sorting, but you can take the adipocyte fraction. And if you look at this adipocyte subfraction, you can see that it segregates in multiple different subtypes of cells. And that we are obviously quite interested to understand which subtypes do they resemble in mice. And actually, if you look at this, so this is a gene expression pattern that shows the different cell types. Here are the adipocytes, and you can see here that most of the adipocytes are quite similar to this population 4, which we identified in mice, which are thermogenically not very active. You can see here, this is the human stain of the adipocytes. And you can see here both the human population 1, but also other subpopulations express high levels of these P4 markers. So given the shortness of time, I can't really go into the function of these cells. I'll just close with this slide here. So I think we have to review our idea of the adipose tissue depot. We have clearly multilocular and unilocular different cell types, and data that we also generated suggests that these have regulatory functions and might be different in thermogenesis. And I think the future will be, we will need to study a lot of these cell types to understand what these different states do and what they contribute to physiology. I just quickly move to one last slide because I want to acknowledge the person who actually did the work. And this is Wenfei Sun here, a very talented PhD student in the lab who is now a postdoc and continues to work in single cell technology to promote this. With this, I'd like to close. Thank you very much for your attention, and I'll try to answer any questions that you might have.
Thanks, Christian. Wonderful talk. We already have some questions coming in. Please use Slido to ask your questions. So the first is, Did you look at the interaction from other cells, such as fibroblasts or T cells with adipocytes, regarding the maturation of adipocytes or mitochondrial activity?
No, we haven't done that yet. So far, we have not done looked at the non-adipocyte cells in the mouse model system. We have done this in the humans because we had to, and as you can see, there's a lot of immune cells in there. We didn't yet have the time to do it, but it's definitely on the agenda. Although I have to say, Peter Tontono, for example, published a very nice paper on interleukin 10 secreted by certain immune cells that interact with the adipocytes to influence adipogenic function. So there's other work on this out there, and more people are looking at this interaction. But our lab is a bit less immune-centered, I have to say.
Okay, thank you. Next question. Does the CYP2E1 population come out during in vitro differentiation? Are some pre-adipocytes primed to give rise to this population?
This is an excellent question, and I would have liked to show this. Due to time, I cannot. Yes, if you do this, you actually recapitulate this heterogeneity in the cell culture plate. You can stain for CYP2E1 adipocytes, and you will see that around 15% of in vitro differentiated adipocytes are later CYP2E1 ALDH1 event positive. So they must have a prime precursor. I'm not saying they have a specific precursor. We have no information on this, but there must exist a prime precursor that generates these cells for whatever reason we don't know yet.
Thank you. Next question. Do you see similar heterogeneity in young versus old?
It's a wonderful question. We haven't done it. I just half an hour ago had a discussion with an aging researcher. We will receive human and mouse adipose tissue in the next few weeks to do exactly that research, but I have no data on it so far.
Okay. Did you see P4 cells in Fluidigm dataset?
Yes, they were called C3 then. So this was actually, if you remember, in the Fluidigm dataset, we had this one very overlapping, and then there was the one C3. This was a bit on the side. And this is actually the same as the P4. So we found that population first in the Fluidigm, and then we confirmed it in the 10X platform. It's exactly the same markers. It's the only population that really segregates. The rest are much less segregated, as you see, probably rather cell states than cell populations.
Okay. Next one. Would you suggest that mice have a larger adipocyte heterogeneity than humans? Or do you find only the single population of adipocytes in human due to sampling reasons?
Sorry, I was a bit too quick on this. Actually, it's quite the opposite. In humans, the heterogeneity seems to be larger. I don't know if it's states or populations, but in the human, our clustering revealed 8 different clusters of adipocytes taken from the neck region in the subcutaneous area. In the mouse, we never found more than 4 or 5. So if at all, the human one is more complex than the mouse one. But they share a certain degree of similarity to the P4, but they cluster independently. So we really don't know yet how big the heterogeneity in the human is. We have to address this.
How does the CYP2E1 populations look at spatially or in different depots of that?
So far, we have looked only mainly in the intrascapular, but spatially, I showed you one picture, right? It appears at the edges of the brown adipose tissue, but it appears in the center. They are unilocular and multilocular. I really haven't found a good explanation yet for the location of these. So we have done thousands of slides, and I still don't understand it. It seems to be random to me at the moment. I'm sure we'll find an explanation at some point, but neither can we assign it to vessels or to neurons. So we don't know yet, actually, to be quite honest. And in humans, it seems to be completely interspersed into the regular depot.
Mm-hmm. One last question. Do you see sex differences in humans?
What? Sorry, can you repeat this?
Do you see sex differences in humans?
Unfortunately, the human data set was most exclusively females. Didn't say this, and only one male in that data set, and we cannot make any statement on this. Based on the 1 versus 9, it seems to be, but this is just the numbers. I don't want to make a statement, so we'll have to add more human samples to this to generate that information.
Thank you very much, Christian. So with this, we conclude this part of our webinar, and we're moving on to the lung network presenter. It's Dr. Nicholas Banovich. Dr. Nicholas Banovich is an associate professor at the Translational Genomics Research Institute, TGen, in Phoenix, Arizona. He received his bachelor's degree in anthropology from Arizona State University and his PhD in human genetics from the University of Chicago. Dr. Banovich's research is focused on understanding the mechanisms underlying gene regulatory changes and how this contribute to complex disease risk, outcome, and treatment response. He has expertise in identifying genetic variation associated with gene regulatory changes and performing integrative analysis on multiple genomic data types. Over the past 4 years, Dr. Banovich has used single-cell genomics to profile regulatory changes at cell type resolution. His work spans a number of disease areas and tissue types, including CAR T-cell therapy for brain tumors, multiple myeloma, and pulmonary fibrosis. And his work is supported by NHLBI, NCI, and the DoD. Dr. Banovich is also a contributing member of the Human Cell Atlas Lung Biological Network. Dr. Banovich, please go ahead.
Thanks for the introduction, Ellen, and good morning, everybody. It's really a pleasure to be presenting on behalf of the Human Cell Atlas Lung Biological Network and sharing a little bit about the network and then about some of our research. So I just want to start by highlighting some of the aspects that I think are really exciting and cool about the lung network, which if you're going to go into the breakout session, some of this might be repeated, but since half of the people here will be going to the Adipose session, I just wanted to give a brief overview. You can see this kind of circus plot of the world here on the right-hand side that shows where many of the contributing members from the lung network are coming from sort of all over the world, representing huge swaths of these different countries. And then on the left-hand side here, I've got an author list from one of our recent publications that is just highlighting the sort of trainee and sort of staff members, not the main PIs involved with the research, but you can see just how many sort of individuals are contributing to this work. And, you know, one of the really exciting things over the past year of being a part of the HCA lung network is, you know, we really took up the mantle of the moment, right? And given the crisis around COVID-19, the leaders of the lung network really quickly saw that we were in a unique opportunity to pull together, share data in really what I would say is unprecedented ways, and try and make some meaningful impact about our understanding of COVID-19. And so many members who had both their own sort of data sets and own individual research interests all pooled together very quickly and put out some of what I would say were really some of the early foundational analyses around where some of the key entry factors and key SARS-CoV-2 related genes were expressed across cell types and across tissue types. And I just want to point out here 3 of the sort of key papers that came out of this work. And so one paper that was published in Nature Medicine, one paper that was published in Cell, and one paper that is right now a preprint but has been accepted for publication. And what I want to highlight here is each of these papers really contributed some really incredibly impactful insights into how SARS-CoV-2 might be infecting cells and what cells it might be infecting and where it might be gaining entry. But I also want to highlight that the 2 papers that were published in Nature Medicine and Cell Press were both published in April of 2020. And as well as this preprint was actually posted in April of 2020. So I think it gives everybody here a sense of how quickly the lung network organized and got together and pushed out this research, you know, in what was sort of less than a year ago, although feels like about 20 years ago. and so, I'm really proud of the work that's been done, and really honored to have been, a small part of this research. And so, kind of continuing on this theme, I'm going to talk about a little bit of work that my lab led, again in collaboration with the larger lung network. Those early papers really focused on the expression of these key COVID genes in healthy individuals. My lab has a strong focus on pulmonary fibrosis and other chronic lung disease. And we wanted to see if we could take a similar type of approach that we'd taken with these other HCA papers, and understand if there were molecular features within individuals with pulmonary fibrosis and chronic lung disease that prime them to either be more susceptible to infection or have worse outcomes when they were infected with SARS-CoV-2. And so what we did is we took data sets from 4 different papers. One paper from my group, another paper from Naftali Kaminsky's group, another paper from Sasha Mishran's group, and then a paper from Robert Lafayette's group, that all had profiled both healthy individuals and individuals with chronic lung disease, either using single nuclei or single cell RNA-seq on the 10x Genomics platform. And again, I want to highlight that all 4 of these papers, the sort of lead or co-senior authors on these papers were all members of the HCA lung network. And so we took the data from all 4 of these papers, integrated it together into one big sort of meta-analysis. We added in about 30 additional individuals that we had sequenced with single cell RNA sequencing separately in my lab, and performed what I think are a set of kind of cool and interesting analyses. And so before I go any further, I just want to highlight, so it's really clear, none of the individuals in this study were actually infected with SARS-CoV-2. This is just looking at individuals who were either healthy controls or who had chronic lung disease, and looking at the expression of SARS-CoV-2 related genes in these individuals to see how they might be modified with this sort of chronic lung disease. So after integrating all of these data together, we ended up with about 600,000 cells across these 4 studies. So the first thing we wanted to do is just look at the distribution of ACE2 and TMPRSS2 expression across cell types. This sort of expression distribution was pretty much what we anticipated, given the other work that we carried out in these other lung network papers, where we see sort of increased expression in these, a little bit in the mesenchymal cells, and then some in the epithelial cells, and generally fairly low expression of ACE2 with much higher expression of TMPRSS2. And remember, ACE2 here is the receptor that's needed for SARS-CoV-2 to infect cells, and TMPRSS2 is the sort of priming factor. But what we can start to do now is look at this is showing the proportion of cells expressing these different genes across all cells, regardless of disease status. But we can also break this down now by different disease status and see if there's any significant difference in the proportion of cells expressing these factors. And while we weren't able to identify any significant differences in ACE2, we do see significant changes in the proportion of cells expressing TMPRSS2 between disease states. And so for the rest of the talk, these are going to be the sort of 4 disease states we focus on. Control cells, so sort of nominally healthy individuals with COPD, individuals with idiopathic pulmonary fibrosis, which is the most severe form of pulmonary fibrosis, or what we call other ILDs. And so this is pulmonary fibrosis that doesn't fall into the idiopathic pulmonary fibrosis bin. And you can see that we have some significant proportional changes here. And in general, we have much lower expression, or in general, we have lower expression of TMPRSS2 in control cells. And here we're only looking at the epithelial cells. We also see increased co-expression of alternative entry factors along with TMPRSS2 in many of the cell types from diseased individuals. So there's been these sort of alternative entry factor genes that have been proposed in addition to ACE2, like BSG and NRP1. And so here we're looking at cells that are expressing both BSG and TMPRSS2, or both NRP1 and TMPRSS2. And in general, we see an increased proportion of cells from individuals with disease. Now, beyond just these proportional shifts, we also obviously want to look at gene expression changes. And so what I'm showing here on the left is just sort of a binary heat map, where we took a list of genes that were manually curated for being involved with SARS-CoV-2 from the sort of broader literature, which I've got to say was a challenge, because at the time we were doing this, there's sort of new papers coming out every day, and the list just continued to grow and grow and grow and grow. But we ultimately came up with this set of genes that we feel pretty positive about. And what we've done is rather than do a sort of formal differential expression analysis on all of these genes, we just labeled everything either binary upregulated or downregulated based on the log fold change, regardless of the p-value, comparing healthy individuals to diseased individuals. And then we perform a binomial test, and we'd expect if there was no general increase or general decrease of the expression of these genes, that they'd be about 50-50. But what we actually see is the majority of these genes tend to be upregulated in epithelial cells from disease samples, with a sort of significant p-value for this binomial test. And then we can break this down a little bit further. And on the right-hand side of this plot, I'm showing this what's called this module score analysis. And this is an analysis you can perform as part of the Seurat package. And basically, you feed it a list of genes. And in this case, this is the genes that are associated with SARS-CoV-2 entry. And then it compares that set of genes to a null set of genes to see if they're sort of over expressed than what you'd expect by chance. And anything greater than 0 here means these genes are expressed at a higher level than sort of a background set. And so what you can see is, in general, again, we tend to have higher expression of these entry genes using this module score approach in individuals with disease compared to control individuals. And to give you a sort of example of a couple of these, just looking at one cell type, this is looking at differential expression of a couple representative genes just in the AT2 cells. And we focus on AT2 cells here because based on the literature and kind of what we know about the SARS-CoV-2 biology, this seems like a likely cell type within the distal lung that's likely to be directly infected by SARS-CoV-2. And so this is looking at just a handful of genes where we can see differential expression between the control and these different disease subtypes. And you can see that some of these are unique to specific disease states, and some of those signals are shared across multiple disease states.
2 minutes.
The other thing we wanted to do is understand how the expression of ACE2 might be influencing other genes within the cell. And so we performed this correlation analysis where we looked for genes that were correlated with ACE2 in the AT2 cells in this sort of disease state specific way. And we see this really sort of distinct gene profile where there's very few genes that are kind of significantly correlated at a high level with in the control samples, and then many genes that are correlated with ACE2 expression in these different disease states, the highest being in COPD. And because we don't want to leave out the immune cells and focus only on the epithelial cells, because obviously while the virus is infecting epithelial cells primarily, the immune component here is really important. We can do the same sort of analysis and look at the proportional changes in immune cell subtypes across these different disease states. And we see these sort of same types of proportional shifts where we see things like increase in CD8 T cells, CD4 T cells in individuals with disease compared to control individuals. And using the same sort of module score based approach, we can group the gene expression values into these different sort of modules, either looking at HLA type 2 scores, cytotoxicity scores, exhaustion scores, and we can compute the sort of difference here and see again that we see lots of changes in individuals with disease, and that many of these changes can particularly when we look at things like cytotoxicity and exhaustion scores, look like they might, the changes that we observe in the individuals with disease might diminish the immune response to infection. And so we kind of come up with this overall model where, based on the widespread cellular and molecular changes in patients with chronic lung disease, we think that they are likely to have greater susceptibility to SARS-CoV-2 infection, along with a sort of diminished immune response to SARS-CoV-2. So I want to acknowledge, that this work was really led by a fantastic postdoc from my group, Lynn Bowie, and a really talented fellow from my collaborator John Kropsky's group, Nikki Winters. These two really drove this whole analysis and did all the work, and I'm really impressed with what they put together. And then obviously the rest of my lab, and then Jonathan Kropsky's lab, who's kind of my main partner in crime, along with some pretty strong contributions from Naftali Kaminsky's group, as well as Gisli Jenkins' group, our funding and support, and then the HLA or HCA for letting us present this work. I also want to just put a pitch in that my lab's hiring for postdocs. So if you're interested in this type of work, feel free to reach out. And I'm happy to take any questions.
Thanks, Nick. Thank you. And thank you to your colleagues. So the questions started streaming in. The first one is, what could trigger higher expression of entry genes? Higher interferon signaling in disease cells?
Yeah, so that's definitely one of the possibilities that we've thought about, is that it could be higher interferon signaling in the samples with disease, and I think that's borne out a bit by some of the signals we see in the immune cells. So yes, I think that's a possibility. It's obviously hard with these types of studies, when we're doing this sort of like retrospective meta-analysis. We don't have a lot of control over the system to really get at these sort of like cause and effect questions, right? So there's a lot of like cellular remodeling and a lot of complicated processes going on in these epithelial cells from individuals with both pulmonary fibrosis and COPD. And so, it's a bit tricky, I think, to pinpoint it back to one exact thing, but I would guess that interferon signaling does play a role.
Okay, great. Any other questions from the audience? Please drop them in Slack. Nick, anything you wanted to talk about that you didn't get to during your talk? You have 2 minutes.
Okay, yeah. I guess while we wait for other questions, one of the things that we spend a lot of time on is trying to integrate these multiple data sets. So I think we've found in our hands the integration within a data set to be fairly straightforward, particularly using, we really heavily leverage the tools developed by the Satija lab through, Seurat and the newer versions of Seurat. We found that some of the like SCT corrected values tend to do a pretty good job of removing a lot of the batch effects within our own study where we have a lot of control. But since these data were integrated from multiple studies, some of which were single nuclei, some of which were single cell, we did have to spend quite a bit of time on integrating these data and thinking about the integration. And we also had quite a big imbalance where some of the studies contribute many cells and some of the studies contributed fewer cells. And so we had to toggle a bit with some of those integration tools to make sure that the variation that was existing in these data sets that contributed a smaller number of cells was getting captured in this sort of integration approach. And so if you're thinking about doing these sort of big meta-analyses with integration, there's definitely lots of levers you kind of have to pull on to get things to run smoothly.
Thanks. And one last question. Did you compare these data sets with others like lung cancers or patients with metabolic diseases?
We did not. I think that could be an interesting question and something that we had thought about a little bit in the beginning. The reason I think we ultimately veered away from lung cancer is that the cells are just so sort of drastically changed in lung cancers that really entering into this sort of totally new cell state. We were a little bit worried about how this comparison would go, trying to compare our sort of canonical cell types back to these sort of very highly transformed cancerous cells. What kind of information we'd get out of there in this sort of apples to apples comparison that we're trying to do.
Thanks so much. So please do continue asking your questions in Slido, and Nick will get to them within the next couple of days, hopefully. And now, Christine, please tell us how to get to breakout sessions.
Great. So now we are going to be going into our breakout sessions. So just a couple of quick housekeeping announcements. So breakout sessions will take place from now until 12 p.m. Eastern Time. Just a couple notes. You will be muted upon entry to the room. We ask that you please utilize the hand raise function, and breakout session leaders will call upon you to talk. If you do get chosen to talk, please just mute yourself once you're finished speaking. And we will be posting the breakout session Zoom room links in the chat. So please go into your breakout sessions now. And then somebody from the HCA staff will be here for the next 10-15 minutes in the webinar should you run into any issues getting into your breakout session rooms. So with that, have a great breakout session, everyone.